Hi everybody. So I just want to continue our revision series of some of the handouts that we have gone through already, some of those topics. And this one may be thinking about transcription. So I guess even considering the way that we started the handout, we had a nice overview of a transcription bubble, as we said, or the transcription process. We could see this RNA polymerase enzyme indicated by the yellow color, the block here in the image, and we could see our DNA helix, which has this gene that's being transcribed here in different colors. So we spent a bit of time looking at the concept of a template strand, which is indicated here by a kind of a pink-purple strand, and a blue coding strand. And you can see that both of them are separated in the middle of this transcription bubble by the RNA polymerase. We can also see a green strand representing the RNA that's being synthesized. So like replication, new strands are always being made in a 5 to 3' direction, with the 3' end being extended. So it's clear that the 3' end here is actually the part that's going to be extended more and more. But this RNA polymerase isn't very much kind of a hop on, hop off system. It's one that jumps on at a particular location, which we discussed at length with initiation, and it slides along, unraveling the DNA or the gene in certain regions. And then as it's gone through those regions and used the template strand to build the RNA molecule, it then rewinds them back together. Okay, so they snap back together at that stage. Only a small amount of base pairs is actually separated. So I think that gives you an idea, even this image, and it's why I started the handout with this, so it let us just grab some of the features of the transcription bubble before we talked about the process. So I guess the key image that we probably spent a bit of time looking through was the idea first of our DNA goes to RNA, mRNA, and goes to protein. And we were going to focus on these protein encoding genes. Right, so genes that would encode an mRNA, which would then go on to build a protein. And when we looked at a gene, I remember I asked you what a gene composes of, or what it consists of. And a lot of you told me it had the instructions or the code. And we learned a bit more since that it had areas called promoters, and it had areas, regulatory sequences and so on, that are involved or associated with it. So while it does have the coding sequence, it has a lot of other features as well. And you can see in the top there in the gene, we have enhancers, silencers, we have a promoter, which was like the on-off switch for turning on and off a gene. We have untranslated regions, an open reading frame, and then untranslated sequence and part of a regulatory region towards the end. And that gene has regions even that are non-coding, and we refer to those as introns. So you can see them here in the kind of center image. The introns and the exons are labeled. So the exons are the pieces of the gene that have the information to code for the final protein. So now you have an idea of what a gene consists of. So we have approximately 20,000 genes. That's the estimation in the human genome. And each of those genes gets switched on or expressed when the product, the protein, is needed for a certain job or a role. So some of our genes, for example, will be switched on all the time. So we call that constitutive expression. It's where the protein is needed for something fundamental or something that's required all the time. So for example, structural proteins, often like actin, they might be on continued during the process of life, I suppose, of the cell, where other genes are going to get switched on as they're needed and then switched off when they're not. And that's kind of what I want you to grasp, is that even during your working day, even during the time you'll spend listening to this video, certain genes in your body will be switched on in certain cells and not switched on in others, or they'll be switched on and switched off later on, and so on. So we have this dynamic system. Even if we take identical twins, while they might have the same genome, apart from maybe a few mutations or changes that happen due to environmental stresses during their lifetime, but if they have the same genome, even if we get their genes that are switched on, there'll be variations between them. Okay, because we switch on different genes at different times in response to different situations or needs of the cell. So that's the main thing. Genes are going to be switched on or expressed. And what we do there is we transcribe and we translate. So we went over those terms quite a lot because they're very appropriate. We transcribe, meaning we stay in the same language. So we stay in the language of nucleic acids. And then we translate when we want to move to a different language, moving from the nucleic acid language to protein. So with those processes, we're locking in here to transcription. So transcription is the generation of the mRNA from the gene. Okay, we're staying in the nucleic acid language, but we're going to make an mRNA molecule that can leave, in eukaryotes, leave the nucleus and go out to meet the ribosomes where the proteins are made. In eukaryotes, we make this version of the RNA first that we called an immature or precursor transcript. And that transcript needs to be processed before it can be considered mature and be able to leave the nucleus. So what does processing involve? It involves putting a cap on the 5' end, a tail on the 3' end, and removing the introns and fusing the exons together in a process known as splicing. So we're going to transcribe and switch on a gene and make a precursor or an immature mRNA transcript. And you can see from this diagram the different features. And then we're going to build, I suppose, our idea of having a 5' cap on one end, a 3' tail on the other, and we'll remove the introns. And then we'll be able to fold a protein at the end so that we can build our protein structure. And we've talked a lot about that in semester 1, the shape and structure of a function of a protein. We also then, when we build a protein, we might need to add things on, like post-translational modifications. This would be the idea of putting, say, a sugar entity onto the protein. That helps its half-life or its effector activity within in the cell. So building a protein is all coded from the information that's in a gene. So that's the first thing we needed to talk about, okay? So inside in the cell, in a eukaryotic cell, you can see your transcription of the actual DNA happening here, very along the lines of what we've just looked at in the last schematic. But ultimately, when we make our mature mRNA, where the introns are gone, the tail is added, the cap is added, we can then move it out for translation, which we'll look at in our next handout. What I want to just talk a little bit about is here, and what's going on inside in that transcription bubble, like we looked at before. So if we just fast forward for a second to this slide, we got the idea of a template strand and a coding strand. And you remember me asking you which one do you think is more important? And we came to the conclusion in class that they're actually both as important as each other. One contains the code, while the other one is a template that allows us to make an RNA version of what's in the code. So I'll just repeat that. The RNA that we generate uses the template strand in order to make an RNA version of the code that's in the DNA, in the gene. So now we make an RNA messenger that's carrying the information that's in the gene, right? But we use the template strand to do that. So I asked you in class, if you think about it, to draw the transcription bubble with a codon as an example. But if you review this diagram here, you can see that the RNA being made here, ACAU, is that sequence is an RNA version of what's in the coding strand, ACAT. But we use the TGTA of the template strand in order to make that, right? So when we see a T, we put an A G, we put a C, T, we put an A we put a U. So base pairing rules is actually appropriate here as well in this structure. So we know what a coding strand is, we know what a template strand is. Now we know what a transcription bubble diagram looks like from the cover sheet or cover slide. And we've talked a little bit again about how the information goes from a gene to a protein. And we focused in on transcription when a gene is expressed. So there are 3 stages primarily in the whole process of transcription. And we refer to those 3 as, was in this diagram here, we looked at them and we said there are 3 stages: initiation, elongation, and termination. But by the time we come to this diagram, we kind of talked about each one and mentioned a little bit about each. But this was the idea that there were 3 stages in general transcription: initiation, the start; elongation, the middle; and the termination, the end. And if you look at this, I like kind of some features of this diagram, because I like the fact that in initiation, the little green dot here is like a traffic light, green for go and red for stop. And remember, these are start sites or sequences, that transcriptional stop sites, I suppose, or sequences. They're not the same as start and stop codons. We haven't got to translation yet, so just be aware of that. We've seen that the transcription bubble, the RNA polymerase, will separate the strands. And once we have 2 RNA bases stuck together or fused together, we have completed initiation, and we've moved on to elongation, where we will move along the DNA strand, extending the 3' end of this RNA molecule that we're building until we reach this sort of sequence, the termination sequence. And that's probably a good phrase for it, the termination sequence. And at that stage, we will then release the RNA, and we release the RNA polymerase to go back and start transcribing another gene. So initiation elongation, and termination. When we looked at these in particular, we were looking at the idea of the gene again, and how in genes we have +1 as the start, where we're going to start transcribing of the coding sequence, but we focused a lot on promoters. And we started looking at promoters being the engine that switches on and off expression, but it's going to help localize the machinery to the gene to the right place. So promoters play a vast, important role, huge role in switching on genes at the right time. So from analysis of sequences that are in promoters, I showed you that there were things called consensus sequences. So consensus sequences are like kind of almost like the average. So what scientists did is they looked back through, say, prokaryotic sequences and eukaryotic sequences in promoters, and they tried to find out what sequences were common to them all. And they began to find, say, in prokaryotes, they had these consensus sequences at -10 and -35. And remember, +1 was where we started transcription, and now we're in this promoter region where the numbers are negative. And the first one there, -10, was known as the Tata box. And then gradually, when we started looking at eukaryotic promoters, we also noticed that there was a Tata box. It was just further back a little bit in eukaryotes, about 25 base pairs back, as compared to in the previous eukaryotes, much lower distance. And there were other features called CAT boxes or GC boxes in a lot of eukaryotic promoters. But we had this Tata. And we spent a tiny bit of time kind of locking into the role of what that Tata does. So we saw that on the left-hand side here, RNA polymerase, the enzyme that I'm telling you is involved in transcription, does all the work, cannot find the DNA promoter. It doesn't know where to land on its own. So it allows, in general transcription, some of these basal transcription factors. So these are general transcription factors here in green. And one of them is called TF, transcription factor 2, because it's RNA polymerase 2 involved in transcription of mRNAs, and D. So TF2D has this yellow circle, so it's called TBP. So this yellow circle I'm using is Tata binding protein. And once this transcription factor can find Tata in the promoter, as we just saw, all the other transcription factors can also localize to this region. And at this stage, RNA polymerase is able to recognize the collection of transcription factors and know it's able to land in the right place. With the help of ATP, it can start separating the strands before it moves into the elongation phase and until it reaches a termination sequence, the termination phase. So essentially, it's key to maybe look back over the diagrams and the animations that we went through with the initiation process, because that sets up transcription really, really well. And then as we go through the elongation phase, we are pretty much along the lines back to this diagram. And the elongation phase will continue until we reach this termination sequence that's going to dissociate the RNA polymerase and the mRNA from the gene. So it's at that stage that we will now have completed our termination particular sequence, and we have our mRNA ready to go. We now need to look at processing the RNA. So going from an immature precursor into a final RNA transcript. So we have to cap it, tail it, and splice it, as we've mentioned. Okay, so I hope that gave you an overview and a reminder of some of the key things that we discussed during our transcription handout, the first part. In the next video, we'll have a look at the regulation of transcription and also how it is processed. So I hope that video gave you a little overview of the transcription process again and reminded you of some of the key things that we discussed. Transcription is a process where we transcribe a gene into a functional messenger RNA. It essentially will build a molecule, the messenger mRNA, that can carry the information and the instructions out to the ribosomes, so we make the relative protein or the right protein, which will be built in the right way and will form the right shape and function to do its job within the cell. So genes are built, I suppose, of these instructions, but we also mentioned the idea of promoters and some of the landing sites that are in those promoters, such as the Tata box. The process of transcription has three main stages: initiation, elongation, and termination. So basically the start, middle, and the end, as we mentioned. And then towards the end, we kind of focused in a little bit on each of those stages, looking at the molecules involved in the initiation, The extension or elongation phase is where we extend the mRNA, and we saw that there is a coding and template strand, and how we're reading the template strand before we reach a termination sequence. What we have to do now, I suppose, is bring it to the next video and look at the other aspects of the cap, the tail, and the splicing processes that we discussed in class. So I hope you found it helpful. Talk to you soon.